Radiotherapy for cancer patients is based on the principle that one size fits all. Despite the fact that we know a significant minority of patients are more sensitive and more likely to have complications due to the treatment. If we could identify this hypersensitive population with a biomarker, we could treat them with a costly but potentially less toxic treatment, such as proton therapy, while giving a higher dose of less costly photon therapy to the rest of the population to increase their probability of cure. This biomarker for toxicity would be the key for personalized treatment in radiotherapy. Ideally, this biomarker should in practice be easy to detect, cheap and non-invasive. Scientists of Maastricht University found that variations in mitochondrial DNA inherited from our mothers could be such a biomarker. Mitochondria are the engines of our cells producing biochemical energy called ATP. But they also generate waste in the form of reactive oxygen species. Similar to all power plants which produce energy but also waste. The waste produced by the mitochondria is damaging for our cells and can induce cell death by apoptosis. Scientists have found that the effectiveness of our mitochondria varies from one patient to another, where some patients are prone to produce more damaging reactive oxygen species and less energy. This can be quantified by a mitochondrial DNA signature and affects the patient's degree of sensitivity during treatment. With our patented mitochondrial DNA signatures, we now can identify these sensitive patients in an easy and effective way. In practice, we can use any normal cells of the body, from blood cells to even cells found in our saliva. We then isolate mitochondrial DNA with a standard technique. We then sequence and analyze the sample with our proprietary CE-marked software. The crucial element is that mitochondrial DNA is small and easy to handle because there are only 37 genes, compared to the many thousands found in the nucleus. So, any genetic lab in the world which has access to our proprietary algorithm could perform this work. The primary application will be to identify and select patients who could benefit from the more expensive proton therapy treatment. Interestingly, we also found that mitochondrial DNA variation can impact the risk for certain diseases such as COPD. For all of these reasons, we truly believe that our saliva-based mitochondrial assay is the key to realizing personalized radiotherapy for all patients.